Welcome to Bioshumit. Myself Sudipa Nandi will try to make you understand a small topic called spermatogenesis. So let's start. Gametogenesis? What is gametogenesis? This is a process in which cells undergo meiosis to form gametes. As we are discussing today with human gametogenesis, there are two types. One is spermatogenesis and another is oogenesis. So now we will learn about spermatogenesis in detail. What is spermatogenesis? The production or development of mature spermatozoa or sperm from spermatogonia is called spermatogenesis. What is spermatogonia? This is a germ cell. Spermatogonia gives rise to mature spermatozoa or sperm. The whole process is called spermatogenesis. Now the place where the process takes place. This is mainly in testes. And if you know the anatomy of testes, there are two compartments like interstitial compartment and seminiferous tubule. And the main place of the spermatogenesis is seminiferous tubule. Seminiferous tubule. The interstitial compartment contains Leydig cell and the Leydig cell release testosterone. Except this, there is some fibroblast cells and neurovascular tissue in interstitial compartment. And in the seminiferous tubule, there are Sertoli cells and they release inhibin B. Germ cells are also present in the seminiferous tubule. So the main hormone for the spermatogenesis is testosterone. FSH and LH from anterior pituitary play an important role in this. So when the process occurs, it occurs at puberty. At puberty, spermatogonia differentiates into two types. One is dark spermatogonia and two is pale or type B spermatogonia. The dark spermatogonia keeps undergo mitosis and they act as a storage system in the spermatogonia. So they don't undergo meiosis, they undergo mitosis only and store the spermatogonia. And the pale or type B spermatogonia goes into division. What division? The pale or type B spermatogonia, that is 44 plus XY, they undergo first mitosis and they form primary spermatocyte, that is 44 plus XY. And the primary spermatocyte then undergo into meiosis 1. And by the division, they form two secondary spermatocytes. So the spermatocyte will maybe 22 plus X or 22 plus Y. And each secondary spermatocyte goes under meiosis 2. And each secondary spermatocyte gives rise to two spermatid. The spermatid also have 22 plus X or 22 plus Y. And the spermatid matures into sperms. So from that we can understand one spermatogonia give rise to 4 sperms. Now next, this picture also depicts the same which I have told you earlier. The spermatogonia goes into mitosis and this is dark spermatogonia which replenishes the storage system, and this is pale spermatogonia which goes into meiosis. And when the spermatid differentiates into sperm, the process is called spermiogenesis. Spermiogenesis. And when the sperms released into the lumen of the seminiferous tubule, The event is called spermiation. So, in the spermatogenesis, there are two another process called spermiogenesis and spermiation. Don't mix these three terms. Now some important points. Time taken for spermatogenesis is 70 to 74 days, and only for this spermiogenesis the time taken is 10 to 14 days. And when the differentiation occurs, the spermatid structures develops into some sperm structures. What are they? The nucleus of spermatid transforms into the head of the sperm. The mitochondria of the spermatid transform into the middle piece of the sperm. The Golgi apparatus of the spermatid goes into the acrosomal cap, and the centriole of the spermatid transformed into the neck and the tail of the sperm. So till now we have learned the spermatogonia divides and forms mature sperm. So please comment below how I have made you understand this topic. Thank you, students.